Okay, hello everyone, and welcome to the first of the 14 virtual seminars by all of the NAI teams. George, we're really happy to have you lead this off. I tried to look up George's CV before this, but actually couldn't find it on the web to give you the usual kind of rundown on where he got his PhD, etc. I suspect that's not entirely necessary since George is pretty well known to everybody in this community. He is an organic geochemist who's been studying meteorites and organic chemistry on the Earth. And he's going to tell us, of course, about what the Carnegie team is doing. And without any further ado, I'm going to turn it over to George. Actually, one slight piece of further ado, just to remind you all that George's seminar, along with all the others, will be archived on the NAI website, so anybody who couldn't participate in the seminar right now will have the opportunity to see the whole thing in almost its full glory within a few days, and you'll just go to the seminar page on the NAI website. And now, without further ado, George, take it away, please.
Well, thanks, Carl. So we're leading this off. We were called to rise to the challenge of being the first to do so. I'll tell you a little bit. The title of our institute is Astrobiological Pathways from the interstellar medium through the planetary systems to emergence and detection of life. We've been involved, the Carnegie Institution has been involved with NAI for quite some time. We were selected in the first CAN. We were reselected in the third CAN 3. At that time, our institute was led by Sean Solomon. He is the PI for the Messenger mission, and realized that at CAN 5 it was not possible for him to lead this and also be a full PI participant in the Messenger mission, which is historical. So I took over the role as PI. In each iteration, we've changed a lot. So each CAN is very, very different than the one preceded it, partly because our own scientific evolution, and also we found things that worked and things that we thought could work better if we changed things. Currently, we have 24 investigators, 6 institutions. Carnegie Institution of Washington is the lead institution. Johns Hopkins University is a new participating institution. University of Washington has been involved with us from the very beginning with John Barros and his students and postdocs. University of Maryland, James Farquhar continues from CAN 3 into CAN 5. The Naval Research Laboratory with COI Rhonda Stroud continues from CAN 3. And a new participant is East Carolina University, where the COI is Matt Schrenk. There'll be a little bit of delay. I apologize. We'll have to get used to this as we switch slides around. What I'm going to do today, which may be a little different, is I'm going to ask the COIs who represent different areas to describe some of the areas that constitute our institution. We basically have 6 primary themes. One is the studies of physical and chemical evolution of planetary systems. 2, the origin and evolution of organic matter in the solar system. 3 is more of a terrestrial or planetary volatiles component, the origin and evolution inventory of volatiles and terrestrial planets. And linked to that is the geochemical steps leading to the origins of life. And 5 is the geobiological and biological interactions. So there's a real sense of flow from the formation and detection and formation of planets to ultimately biological systems. And finally, task 6 is linked, an astrobiological link, if you will, to our field studies and integrating that with laboratory flight, experimentation and flight instrument testing. So task 2 and 1 are essentially astrochemical, astrophysical in nature, and Alicia Weinberger, who's sitting on my left, will discuss the subtasks there. Tasks 3 and 4 link to fundamental issues related to terrestrial planet formation and early evolution. And Steve Shirey, who sits on my right, will lead through those sections. And then tasks 5 and 6, the geological, biological interactions and the fieldwork associated with this will be discussed by Marilyn Fogel, who's sitting on my far left. So I'm going to turn this over to Alicia, and when this next slide comes up, she will take you through tasks 1 and 2.
Thanks, George. So we're going to start with studies of the physical and chemical evolution of planetary systems. And this starts with astronomical observations that inform our understanding of the diversity of planetary systems and the history of our own solar system. You can see there all of the COIs who participate in these, but you don't have to read them all. I'll talk about them each in turn. So first we'll start with searches for planets, and that begins with my co-investigator, Paul Butler, who's one of the preeminent planet hunters using the radial velocity method for finding planets. And on this slide, you can see a plot from his work showing the number of planets that have been discovered as a function of mass. And there are now about 340 planets known. They're mostly known from this radial velocity method. So that is looking at the motion of a star toward and away from you as it's tugged by a planet that is going around it. The lowest mass planet that's been found is about 5 Earth masses, and maybe one has been discovered in what we would call a habitable zone. So the challenge for the future is to find planets of lower mass and to find them in the habitable zones of sun-like stars. So the challenge there is to get better radial velocity precision. And what you can see here are these blue lines, like this one, that show what the precision that can be achieved in the next now and in the next couple of years is. And that's a 3 sigma of about 3 meters per second. You have to stop and think about that number if you haven't before, because that's like a fast sprint, right? So kind of amazing that you can measure that around a star that is at 15 parsecs or some distance away. So this plot shows stars that have already been discovered to have planets in the orange dots, kind of what the state of the art has been for the last few years. That's about 10 meters per second, 3 sigma. And where Paul is attempting to push the technique over the next couple of years, which is a very consistent 3 sigma of 3 meters per second. And also shown on here in the green and in the purple lines are limits that one could get from astrometric measurements, and I'll come back to those in a couple of minutes. But I think what you can tell from this is that around sun-like stars, the technique that's really going to probe to the lowest mass planets and at interesting separations from the star in the next few years is going to be the radial velocity technique. And this next slide, when it comes up, is going to show a simulation of what radial velocity could do with a precision of 1 meter per second and long telescope runs, so that you can really get a cadence necessary to observe something that orbits its star frequently. And this particular simulation was done for a half a solar mass star, and that places the habitable zone at about a period of 50 days. So this is for a 5 Earth mass planet, and on the top plot you see what it would look like if you actually took data with a precision of about 1 meter per second every day for a year. And then if you did an analysis of those data, you would find that in a power spectrum, the period of 50 days does indeed come up very strongly. And so you could detect that 5 Earth mass planet. Although you can see that that planet produces a signal of about 1 meter per second. And so it's very difficult. This is the cutting edge of what they'll be able to do in the next few years. And Paul will be commissioning a new spectrograph on the Magellan Telescope at Las Campanas Observatory this year that will hopefully do better and do meter per second precision. And another of our co-investigators, John Chambers, is working on advanced computational techniques in order to tease out planets, especially multiple planet systems, from radial velocity data. So I mentioned astrometry as another technique for finding planets. And we also have an astrometric planet search going on at Las Campanas Observatory on the DuPont telescope with a brand new camera that just flashed up there called CAPSCAM. And this is a project led by Alan Boss, although I'm a co-investigator. So if we now start thinking about astrometry instead of radial velocity, that's watching the motion of a star on the plane of the sky as it's tugged on by a planet going around it instead of toward and away from you. And the astrometric signal is increased by having a lower mass star, so that a planet of a given mass makes it move more, and also by looking at stars that are very nearby, because that way their apparent motion on the sky is larger. And in this case, then you're driven to look at lower mass stars and stars that are very nearby. So for solar-type stars, like the ones that Paul Butler is interested in order to get a reasonable sample, like 100 stars, you'd have to go out to a distance of about 30 parsecs from the sun. Whereas to get a sample of very low mass stars, those are the M-type stars or even brown dwarfs, you only have to go out to 5 to 10 parsecs to get 100 stars. And so now a precision of 250 microarcseconds, which is the yellow line in this plot, enables you to detect Jovian mass planets at interesting distances, astronomical unit sort of scales, from nearby low mass stars. And that's the principle of the astrometric planet search, and it should be going on at Las Campanas Observatory for the next decade. So studies of extrasolar planetary systems have already shown that most planetary systems are not like our own, right? They contain planets at a wide range of eccentricities, at a wide range of separations from their central stars. You have low-mass planets that can be further out or closer in. So a fundamental question that we want to, astrobiological question we want to ask is, how did our system come to be habitable? How did we wind up with a planetary architecture that allowed us to have a habitable Earth? And that is the question that's asked by Scott Shepherd, the next of our co-investigators, by studying the small bodies in our solar system. So this slide shows a cartoon of the solar system where we have currently today reservoirs of small bodies, that is asteroids and comets, primarily in the Kuiper Belt, out at 50 or so astronomical units from the sun, and the Oort cloud, much further away. But the important thing is that those bodies came to be where they are and then the dynamical distributions in which they are because of the interaction with the giant planets. And so it's the architecture of our planetary system that resulted in the locations of the small bodies as we see them today. And in particular, there are some bodies that are very hard to understand given what we know about the architecture of our system. And so, for example, Sedna, which is one object that people may call the first detected inner Oort cloud object, or it's the furthest detected Kuiper Belt object, is very difficult to understand how it got in its present orbit. It's in a highly eccentric orbit that takes it only about 70 AU from our sun at closest approach. So there are a couple of possibilities. Either we had an encounter with a nearby star, or perhaps there's an unseen planet out in the Kuiper Belt that modified Sedna's orbit. And so Scott Shepherd's project in the next couple of years is to search for more of these objects with strange dynamical and formation histories, such as Sedna. So he's going to do a very deep, very wide field survey looking for objects in the outer Kuiper Belt. And on this slide, you can see kind of the state of what's known today for small bodies in the outer solar system. Most of these bodies can be explained by having had interactions with the giant planets. And Sedna is in a class of only a couple of objects at this point that really seem to have had dynamical interactions that we can't understand just by the giant planets. And so Scott will be looking for more of those and trying to understand, based on the distribution of those small bodies, what had to have happened in our early solar system. So we can do analogous work to looking at the small bodies in our solar system by looking at the disks around other stars. And that's my interest, but I won't spend the next 15 slides on it, I promise. So here is an image that we took with the Hubble Space Telescope of the disk around another star, HR 4796a. And the dust that's in this system is created by the evaporation or the collisions of planetesimals, the equivalent to the asteroids and comets in our own system. And so when we look at these systems, it's that kind of population of small bodies that we're looking at, and we try to relate that back to what we see in the small bodies in our own system. And in this particular image, one of the interesting things that you can measure, although it's very hard to see visually, is that one side of the disk, this brighter side on the left, is actually slightly closer to the central star than the opposite side of the disk is. So the disk probably has some small intrinsic eccentricity. And one way to get intrinsic eccentricities in the population of small bodies is to generate them with an eccentricity of a planet in the system. And we do know that in most other planetary systems, the planets are in eccentric orbits and not in circular orbits. So furthermore, we know that this system went at least as far as making planetesimals in terms of growing things into planets. And it might be analogous to what the early solar system looked like at a time when there was a much denser population of small bodies than there is in the solar system today. Now on the right, you see the color of this disk, which is very red, and I'll come back to that color in a couple of minutes when I talk about the study of organics in our system. So now you've seen a little bit of the flavor of what we're doing on the diversity of planetary systems and trying to understand the architecture of other planetary systems and of the origin of the architecture of our habitable system. But even if we understood all of that, we would still have to question or try to understand how we managed to get a habitable planet in our particular architecture. And that is tied up with how you deliver the materials to the early Earth that you need in order to form interesting biochemistry, in order to have habitability. And so the second major thrust of research in our astrobiology group is the origin and evolution of organic matter in the solar system. And you can see the co-investigators on this slide who are involved in that project. And this spans now astronomers to laboratory astrophysicists, as I like to call my colleagues, meteoriticists, and organic chemists. So the first thing we can do is ask, in our own solar system, where we can really measure the current day population of small bodies, what are they made out of, and do they have interesting organic contents? And so this, again, is a project that Scott Shepard is interested in, where he looks at the colors of bodies in the outer solar system, in the Kuiper Belt. And this shows, and if you're not an astronomer and not used to looking at color diagrams, basically it shows how red objects are. As you move to the upper right of the diagram, you get objects that are redder and redder. And there's a class of objects that Scott calls ultra-red. And our best idea for how they got to be so red is that they're covered with an organic sludge. And this organic sludge, sludge is not a technical term. This organic material might be similar to the material that we see, for example, on Titan that's been characterized as Titan tholins. And it might be made in interesting chemical ways. You might, for example, bombard ices, particularly methane ice, because it has carbon, with ultraviolet or charged particles from the sun, and create these complex organics on the surfaces of these objects. And so what Scott intends to do is probe the distribution of colors in the outer solar system along with what the ices are that are actually present on the surfaces of these bodies, to try to understand out of what ices, if in fact it is ices, you make this ultra-red material that we observe, we think, to be organic rich. And that ties in very nicely to what I try to do around disks around other stars, where I try to understand what their colors tell us about their compositions. And so here is shown on the right a number of different disks around stars, so 6 of them, that have different colors. And in black is the one that I showed you earlier, actually, HR 4796. And that's also what's shown on the left in black. So that particular disk has a color which is very similar to the color of red outer solar system objects. And shown in red, overplotted with it, is a centaur, which is basically a Kuiper Belt object named Pholis. So the similarity in color that we see between some of these red exosolar disks and the red colors that we see in the bodies in the outer solar system, we think that the same astrophysical planetary processes are at work in both kinds of systems. And so we'd like to make the analogy from these red organic rich objects in our system to the dust and bodies in other systems as also being red and organic rich. And so Scott's work on the solar system will continue to inform what we can say about the composition of the material around other disks, as I try to ask the question how many other planetary systems were capable of forming habitable planets and seeding them with the carbon that was necessary for life. So in that vein, it is small bodies that bring the volatiles and organics that are interesting for life to young planets. And a major thrust of our astrobiology group is to actually investigate these kinds of materials in situ. So we can do these remote sensing observations, and we can look at outer solar system bodies, we can look at other disks, but there's nothing like actually looking at the organics in your laboratory and trying to figure out how they came to be as they are. So there's a lot of state-of-the-art instrumentation that's being brought to bear on the problem of meteoritic and cometary organics, both at the Carnegie Institution and at the institutions of our co-investigators. So this is a slide from Rhonda Stroud showing an in situ picture of organics in a meteorite, and that's what's on the left here. And it turns out that one of the mysteries of organic matter in meteorites is why much of it is found in these little nanoglobules, these tiny isolated hollow spheres. And presumably that unusual structure for the organics has something to say about how the organics were formed in the meteorite parent body. But it's only by studying the organics in situ that you can really be sure that the shapes and nanoscales that you're observing actually reflect what the parent body looks like and not some processing that happens in the laboratory afterwards. Although on the right is isolated organic material, which does in fact still show one of these globules. So the real question is, this organic material in meteorites is insoluble organic matter, which makes 70% or even close to 100% of the organic material in meteorites. Where did it come from? What is it? And how did it get to its present form? So a number of different analytical methods are being brought to bear on these questions. And in the next slide, I'm going to show you how a couple of them interrelate. And that is looking at the isotopic and compositional composition of meteoritic organic matter in situ. So shown in the color on this slide is a NanoSIMS analysis of the isotopic composition of the organic material. And you can see that rather than being isotopically homogeneous, there are hotspots, deuterium-enriched hotspots, nitrogen-enriched, nitrogen-15-enriched hotspots. And so despite the fact that the organics have undergone considerable processing in their parent bodies, somehow they've managed to maintain these very small-scale inhomogeneities. And that is another key question that the group here would like to answer about how the organics formed and were processed in meteorites. So once people like Larry Nittler, who's actually sitting behind me, have gone ahead and analyzed the isotopic composition of the materials, Rhonda Stroud can go in and actually extract little segments of the meteorite that are known to have these isotopic anomalies. And so what you see in the grayscale underneath is an electron microscope image showing the little segments that she can go and actually cut out with an ion beam. And you can see that those little segments run right through those inhomogeneities, those hotspots in the isotopes. So once those pieces are extracted, then even more analytical tools can be brought to try to understand what that says about the kind of organics those hotspots are located in. And so here you can see on the lower left an actual image of the area of that meteorite that's been extracted with the enriched hotspots along it. And on the right, X-ray spectra that show the molecular form of the organics that contain those hotspots. And so together, the idea is to use all of these techniques to ask how it is that you formed this insoluble organic matter, how you incorporated these inhomogeneities into that matter and preserved them through whatever alteration was happening on the meteorite parent body. So once you've done this, you then still have to come up with an overarching theory of the organic matter. And it's pulling together all of these different results from the different analytical techniques that will really result in a comprehensive understanding of the organics and hopefully reveal how it originated. So this slide shows some of the additional techniques that George is applying to the organics, both those extracted from meteorites and from the Wilt-2 samples, and also reproducing them in the laboratory to understand what the physical conditions are that may have produced the organics that are measured. And so On the left, for example, you can see some results from typical meteorite insoluble organic matter compared to cometary matter, and they bear many similarities. In the middle, you can see a two-dimensional nuclear magnetic resonance imaging that shows the kind of structure that organic matter takes and implies that it originated in simple sugars or in sugar-like structures. And in the laboratory, George can take formaldehyde and through to sugars and do it under different temperatures and under aqueous conditions and produce a polymer, an insoluble organic polymer, which closely resembles in its spectrum the actual organic material that is found in meteorites. And it appears that way not only in its spectrum and its composition, but also in that image in terms of the ability to form the small spheres like what I showed you earlier. And so there's the possibility that all of these organic solids originated from a set of chemistry starting from very simple precursors such as formaldehyde, but that's a conclusion that remains to be proven and is a thrust of George's work over the next few years. So that wraps up the first two tasks of our astrobiology group. So I'm going to turn it over to Steve Shirey now to take off from there.
Well, I'm a new investigator, to this team, and I got involved because I have a lot of expertise in Archean geochemistry and in interaction between hydrothermal solutions and ocean ridge systems, and I've been really worried over the last 10 or 15 years on the formation of continents, and of course forming a stable continental platform makes a platform for life that's in the photic zone. And so all these processes happening in the early Earth are very, very key on when our Earth does become habitable. What I'm going to talk about today are items 3 and 4 on this list here, which is really having to do with the rocky planets and what our investigative team is doing. The first is on volatiles, the carbon, hydrogen, oxygen, and nitrogen volatiles in planetary interiors, and the second will be on primary carbon in Martian meteorites and terrestrial analog systems. So let me start with our investigator, John Chambers, because this is where, you'll see in the next slide here in a second, where the original inventory of volatiles comes from. John is doing simulations of giant planet formation that involve the considerations of migration of planetary cores inward, and finds that when you involve migration in these calculations, you can see in a plot here the most massive bodies versus the total disk mass, you produce more habitable or more gas-rich rocky planets that will allow you to to start with perhaps a better inventory of volatiles. And the question of whether our inventory of volatiles is a primary one or volatiles are delivered after is really the starting point for the study of planetary volatiles that we're concerned about next. The next item that I'll talk about is a standalone project, the MESSENGER project involving Mercury, and work that Sean Solomon's done over the years on Mars. This involves investigators Sean Solomon and Larry Nittler. Larry, again, back to my left here. I feel funny talking about it with him sitting behind me. But the key question here is when we look to the extreme limit of the smallest rocky body in our solar system, how does that place a limit on the kind of conditions that we have on Earth for volatile our volatile inventory? So the key point about the MESSENGER is it's a standalone project, but it brings a great intellectual diversity in the thinking that helps us just add to the richness of the kind of discourse we have here within the team. Another important thrust is trying to put some limits on the solubility of water in magmas, and especially in lunar magmas. This has been the work of team member Eric Howry and his collaborator Alberto Saul from Brown. And they've come up with a very interesting result by doing ion probe work on lunar glasses. And that result has given us a much a theoretical or an empirical look at the water content of the moon, and says that it's much higher than we originally thought. And that's a very interesting result because during the giant impact, it means that we don't have to really dry out the silicate material that was splashed out of the Earth's mantle and formed the moon. And it also bears into what the final solubility limit of water might be in these kinds of processes. My own work is involved in looking at diamonds and the importance of early deep-seated carbon on the Earth. In the next slide here, we'll see some of the work we've done on continental cratons. It takes a while for these slides to refresh. Continental cratons on Earth are the storehouse of the oldest rocks, and you see in this particular slide here a cross-section through a craton on the left from the bottom, where we have a piece of mantle that's stuck to the craton and is ancient. And with kimberlite eruptions through this deep portion of the mantle, we see diamonds, eclogites, and peridotite that are brought up in one particular eruption, any particular eruption. And the diamonds themselves, the importance of those is those are the oldest, deepest carbon-bearing phases we can find, and the oldest phases that will give us some idea of where carbon came from during early tectonic processes on Earth. Now, if you look at this cross-section through cratonic mantle, what you see is the areas where we can get good information. And diamonds are also being now recognized from below the lithosphere. So we really have deep-seated carbon that we can study through a look at natural diamonds, not only from, do we have a mouse here?
There's a mouse here.
Mouse. Not only do we have diamonds that are part of the lithosphere here, these ancient root zones right here, but we have now diamonds that come from the transition zone and represent deep mantle recycling into the transition zone. And that's a very exciting new find that we're going to be trying to capitalize on in the work. Diamonds are important because they span a great age range across the type of changes that we've seen on the Earth. And so in the next slide, you'll see a plot here of diamond age ranges from different mines. The mines are over here on the left. These are various localities. And then the age range of diamonds that have been found from those particular mines are these black bars. And you can see that there's quite a range from the Proterozoic all the way through into the Meso, even to some cases the Eoarchean. And that not only spans the range of major crust formation on the Earth, but also spans the change in from reducing atmosphere on the Earth to oxidizing atmosphere on the Earth. And so when we go back and try to reconstruct where these carbon-bearing fluids came from, we do have a chance to really look at a tremendous range in the geological evolution of our entire planet. The way this is done is by determining the age through looking at mineral inclusions in diamonds. And in the next slide, you'll see some pictures of the types of inclusions. I won't go into a lot of detail, but there are two main types of inclusions, silicate inclusions and sulfide inclusions. And we can work on each one of these. We've done a lot more work recently on the sulfide inclusions because we can analyze individual diamonds. But one of the things we do is to not only get the age from the sulfide, but we look at the carbon isotopic composition and the stable isotopic composition of the sulfur. And from that, we can try to determine whether the source of the carbon was biotic or abiotic. And in some cases, we can also look at the nitrogen isotopic composition of trace nitrogen that's in the diamond. A really interesting result that was obtained by one of the team members, James Farquhar, was published in 2002 in Science. And in a diamond from Arapa, something that was stored at 150 kilometers in the lithosphere, he discovered mass-independent sulfur isotopic composition differences from the normal fractionation that can only have come from photolytic processes in the stratosphere before we had shielding oxygen to block those kind of isotopic changes. So the striking implication is that the sulfur from volcanic eruptions up into the stratosphere in the Archean was able to these eruptions produce sulfur that was able to have its isotopic composition changed. That sulfur was then brought down into sediments that were then subducted down into the lithosphere where they became part of diamond-forming fluids. So this is prima facie evidence that we have surface carbon as inclusions in diamonds. Now, one of the nice things that's been added to the team is by new investigator Bjorn Meesen. And Bjorn Meesen's expertise, in the next slide here, is in studying the solubility and solution mechanisms of various volatile species, depending on what the redox state of this particular experimental procedures are. And so here he's got some experimental studies of volatiles in high-pressure silicate melts. And the interesting thing on the left here is you can see at different pressures here, pressure is increasing with a bunch of spectral lines here, Raman spectra. The higher pressure lines are at the top and the lower pressure on the bottom. You can see a change in the absolute concentration or the relative concentration of methane, hydroxyl, hydrogen, and other species. And what's interesting about the work is that not only is he able to look at the change in the composition of the volatile species, but there's also a carbon isotopic composition change that seems to be coming out depending on the type of oxidation state and the type of composition and structure of the melt. So over here on the right, you can see a del C13 change as you change the non-bridging oxygen to silicon ratio or the melt composition. Bjorn is also working in nitrogen and at different oxygen fugacities. And what you see in the next slide here is, again, a Raman spectra on the left, showing the range of species ranging from ammonia to hydroxyl to nitrogen dimers, and that the bulk composition of those nitrogen species changes with oxygen fugacity. So does the solubility limit for nitrogen, and there's a tremendous change depending on oxygen fugacity. In all cases, nitrogen increases with increasing pressure, but the solubility goes way up at reducing conditions. So these are some of the fundamental questions that this experimental approach will help us get to try to understand how volatiles are stored and how they cycle through the Earth. Another new team member, Alex Goncharov, is taking this to extreme conditions, and here we have some examples of how he's looking at more extreme conditions, not so much at the type of things Bjorn was looking at, where he's looking at the actual speciation and melt structure, but really looking at what kind of species exist at extreme conditions, and also modeling it with some molecular dynamics simulations, which you see on the right. And finally, we get to the work of Andrew Steele, a long-term NAI team investigator here. And on the top of this series of photomicrographs, you see here are these top three panels, A, B, and C, are natural samples of a peridotite from Svalbard, from the Bokfjord region, and the bottom two rows are two Martian meteorites. And what he's doing is trying to look at the storage of macromolecular carbon and the hosting minerals for macromolecular carbon, and try to, from the terrestrial localities, look at what the analog conditions might be for the storage and how this macromolecular carbon is stored in the various minerals in these particular meteorites. And with that, I'll turn it over to team member Marilyn Fogel.
No, that's George.
Oh, to George, I'm sorry.
Well, actually, I thought you were going to cover this, but I can understand why you don't want to, so I'll be very brief.
Oh, okay.
No worries. So it turns out all of these studies of essentially the delivery of, the storage of, and the release of volatiles, needless to say, feeds into fundamental issues related to potentially a connection between the geochemical evolution of the planet and the emergence and origins of life. This is a smaller task in the sense it's myself and Bob Hazen and Dmitry Sverogensky from Johns Hopkins University, the new co-I. My interest has long been and continues to be the extent to which mantle-derived carbon and perhaps hydrogen, either directly or through serpentinization, could be used in the presence of various transition metal sulfide catalysts to drive a complex reaction network. And so these are a series of experiments that we do under various temperature, pressures, and fluid compositions with appropriate catalysts, and we simply try to follow the chemistry, if you will. Of course, this is just a chemical reaction network. It works under fairly dilute conditions. So Dmitry Sverogensky and Bob Hazen have been intensely interested in how mineral surfaces may play a fundamental role in selecting out of a relatively dilute if you will, a soup of complex compounds, and not just select them, but maybe perhaps even allow them to do things. So in this slide that's finally popped up, you're looking at a very recent study, came out of Dimitri's work with actually 2 NEI-funded postdocs looking at glutamate attachment on root teal. Dimitris Verjensky is the expert on surface thermodynamics, surface chemical thermodynamics, and he has come up with some very sophisticated models to understand how various organic compounds would ligate to surfaces based on pH and electric double layer effects. And on the left, you see experimental results that his 2 postdocs have performed, doing very careful titration studies. And you see, and without going into any detail, that in fact, it looks like Dimitri's getting this right. And so this is just the beginning of a 5-year project to sort out some very, very complex phenomena on mineral surfaces. So with that, I'm going to hand it over to task 5 and 6, Marilyn Fogel, and I will get this thing to go find Marilyn. Here she comes.
There's Larry.
There's Marilyn.
Oh, okay.
I'll get you. And here's a laptop for you. Yeah, I'll get you in there.
Okay. Good afternoon. One of the, I think, the strengths of our biology program here at the Carnegie is that we have a very close association. Organic studies that are derived from non-biological means. And one of the major thrusts that we've had, at least for the past 5 years, is to be able to distinguish biologically derived organic matter with abiologically derived organic matter. And most of us on this team who are considered on the biology end also participate very heavily with Steve and with George in understanding the properties and characteristics of organic matter in terms of isotopes and composition and how they look under microscopic methods. So we begin the strict biology session by studying life at extremes. And for the third time, we're working with our long-term colleague from the University of Washington, who is John Barros. Probably everybody knows John. And the studies that he's particularly interested in completing for this project are looking at and assessing the importance of hydrogen-supported microbial biofilms. John has been studying biofilms and looking at hydrogen growing microorganisms for a number of years. And if you know John, he always has some very interesting questions that he'd like to answer. And the main one he's going to address in this proposal is, you can see on there, why biofilms are important to the origin and evolution of life. And in this question here, he's thinking of looking at what he calls pre-cells and the Earth's earliest network. And at the moment, he's very enamored in studying horizontal gene transfer, the transfer of genetic material from one species to another species. And as part of this work, there are 2 different kinds of projects on here. One is with a new student, Billy Brazelton, who is a student at the University of Washington, who will be studying the Lost City carbonate chimneys, and we'll go into the differences there. And former student Matt Schrank, who was also a postdoc at the Geophysical Lab here and is now a colleague at East Carolina University. Matt will be looking at the comparative structures in sulfide chimneys. So if you look at the next graphic that comes up here, and the very interesting comparison that we see is between, on the top panel over there, studying hydrothermal vents in magma-hosted systems where the pH is low, temperatures are fairly high, extremely high, I would say up to 400 degrees. There are ample carbon sources, catalytic minerals, and I would have to say that that sort of environment is something that, together with the microbiologists, dovetails exactly that work with George Cody has done over the years with George and his colleagues. Down below on the lower panel there is the work on the peridotite-hosted hydrothermal systems from Lost City. And you can see their temperatures are more moderate, pH values are very high. This work dovetails with some earlier work we did on the last can at the Cedars microbial system out in California with Penny Morrill. And in general, what our goal here is to compare the two different types of hydrothermal systems and assess the different type of microorganisms that are found in there. What you see on this next slide is something that on the upper right-hand corner over there, there's a great picture of, John points out that greater than 80% of those cells are a single species, Methanosarsonales. And what John is getting a handle on here is that within the biofilm there, that individual cells are conducting different and very specifically segregated types of metabolism, showing the importance of a biofilm rather than single cultures. Down on the lower left hand, you see a diagram of the number of operational taxonomic units. That's what OTUs are. And basically, on the one scale, you can see that the preponderance of organisms are the one species, but if you trace that out, you can see that diversity and rare cells are something that is a feature of these kinds of ecosystems. And I think this slide came from also a collaboration with Julie Huber, who is now at MBL in Woods Hole. She was a student supported by the NAI during Can 1, I believe, and also an NAI postdoc throughout this time. Matt Schrenk is continuing to work on biofilms and mineral catalytic systems. What you can see over here is he's going to compare laboratory cultures with biofilms that were formed in situ on vents. He's been the last couple of years doing experiments, trying out novel culture mechanisms. As you know, some of the challenge in the microbiology field is that you're only able to actually culture, I think it's less than 1% of the organisms that potentially grow within any one environment. And this is some work from Vulcano, Sicily, where he's collaborating with Jan Amund and other scientists who are also part of NAI. And I should note that Matt is now at East Carolina, where our former astrobiology chief, John Rummel, is now the director of that. And we would imagine that this is a school in which he hopes to attract a lot of undergraduates to the field of astrobiology. The other kind of extreme environments we're studying is the effect of pressure on microbial growth. And you can see on the right-hand side there cells that were subjected, this is E. coli, that were subjected to greater than 100 megapascals of pressure, probably going up to, well, you can see up to 400 megapascals here, which is at the limit of our hydrothermal reaction vessels that we have here. And what you can see on the left-hand panel is that while most of the E. coli is killed at these extremely high pressures, experiments with halobacterium, a salt-loving bacteria by Adrian Kish, who is a new postdoc here at the laboratory, show that, as it put on there, not all microorganisms alike. Adrian is working on molecular biological methods for determining what the actual biochemical underpinnings are for the ability of halophiles to survive the very high pressures. And Patrick Griffin, who was possibly going to be a real graduate student, he's a half graduate student right now at Johns Hopkins in the fall, is right now he's doing some stable isotope experiments with hydrogen isotopes in my laboratory. We have a lot of field work planned for this team. Adrian Kish, microbiologist, and Mihaela Glamochlia, who is also a postdoc, plan to go out to White Sands National Monument, where there is one of the largest aeolian sulfate deposits out there in terms of lake beds and evaporites. Adrian as the microbiologist and Mihaela as a budding microbiologist, geologist, geochemist are going to go out there together. It's a very interesting team. They work with Andrew Steele. They're going to study the geology, put this in context with the microbiology, and bring back a number of samples to work on both through a Mars analog perspective and also microbial biogeochemistry. Some other work that has been ongoing here in the laboratory is the AMAZE expedition, Arctic Mars Analog Svalbard expedition. This is work, I chose this picture here. You can see the research vessel that we use. And this was up in the, at 80 degrees north latitude in Svalbard, and this was probably the most desolate region where I've ever been. And we have a team of people here studying life in ice, determining, that's headed up by Jen Eigenbrod, who was part of the Goddard team. On this diagram over here, we're also studying how microorganisms respond to cold environments and environmental parameters. This is a graph that was, it's actually hand-drawn by Verena Starkey, who was a graduate student at the University of Maryland working with Andrew Steele and Frank Robb. And what she's studying here is the most northern hot spring that's found in a terrestrial environment, going from a very a wet environment up here where the spring is actually flowing to where it dries out, and eventually there's dry terraces here which are cooler and drier, and of course the organisms are immobilized in there. And she's studying the microbial diversity in here and how the organisms adapt to cold, dry environments, which is something you would find on Mars. Now, we're going to switch from this to studies on biosignatures. We've been working on this for a while. These are figures of stratigraphic water columns. This is work by Dominic Papineau and colleagues, and Dominic has been going around the world collecting Precambrian rocks of various types, and something that he is especially interested in is the linking up the phosphorus cycle with the carbon, nitrogen, and sulfur cycles. So what is depicted here are two different scenarios of potential water columns. On the left is a scenario where phosphate dominated the ocean environment, and there were anoxic waters that occurred fairly high up in the water column, even extending into the euphotic zone where there was light. Versus on the right-hand side of the diagram over here, this is in a non-phosphate domain where productivity is still a high productivity area, but the anoxic boundary zone is much lower in the water column. And looking at the biosignatures of nitrogen, linking these with sulfur and carbon is one of Dominic's goals in this project. We dovetail very nicely with James Farquhar. James has been a collaborator with us for the past 5 years, and he's continuing. He's probably best known for his discovery of mass-independent isotope fractionation of sulfur isotopes. He has been studying with his students, particularly Dave Johnson, who is now up at Harvard. So he's created a Harvard professor, which I think is pretty good. mass-dependent fractionations, and what he proposes to do in this particular can is to study the small mass-dependent fractionations that have been catalyzed with particular cultured microorganisms. So he's going to do a lot of culture work from that. A third biosignature study is being carried out also by Dominic Papineau in collaboration with Brad DiGregorio and Rhonda Stroud, who are at the Naval Research Lab, Jinhua Wang, who was one of our NanoSIMS people here, myself, and George Cody and Andrew Steele. And what you see on here is a number of tiles. Dominic likes to show tiles over here. And he's studying, these are the Quelea rocks, QP rocks from Greenland. They're about 3.8 billion years old, and the origin of the carbon, which can be seen in here as graphite right here and here, also in here and here. These are Raman scans over here looking at the various types of carbon. And Dominic, who is also funded by an exobiology proposal, is studying these graphite bands, inclusions. They're not inclusions. They're coatings and vaginations. They're found in a variety of different places within this rock. And he's testing the hypothesis as to whether or not this carbon may have originated through biological processes. The work is controversial. People argue about the type of the rock. Was it sedimentary? I think people have decided on that. It's very highly metamorphosed, and he is having to deal with the fact that he's looking at organic carbon that's highly metamorphosed, and in fact there are very few examples, almost none, I would say, and if we have them, it would be work of Andrew Steele's, looking at similar mechanisms as to what has happened during metamorphism of non-biologically formed complex organic matter. So Dominic has his work cut out for him, and he proceeds to use a lot of the equipment that we have here in the Naval Research Lab. Going off to the side over here, Bob Hazen, who has just recently published quite a large review paper on mineral evolution. It's highlighted on our website. And you can see from this diagram over here that it's his hypothesis that when life evolves on a planet, the number of mineral species increases dramatically. We know what's on Earth, and he has on the lower right-hand panel greater than 4,300 mineral species. Shortly, when MSL actually flies, it will host Kemin, which is going to be able to do a bang-up job of the mineralogy on there. At the moment, he's estimated about 500 mineral species have been found on Mars. I'm not exactly sure where the Venus number came from, but this obviously is known for the moon from the rocks that we have in hand. And we thought about this one, and I think it's really something that's interesting as we send things out in space and fly and get data back as to whether or not we have a biosignature involved on there. Nearing the end of this, we're actively involved in testing instruments that are a couple of them that will be flying on MSL through an A-STEP program. Andrew Steele is the principal investigator on this program. Many of I go on this field work all the time, and many of our students and postdocs attend these. What I have on this slide is showing the JPL's CliffBot rover over here, and we as a scientific group have learned to do science with a rover. We also have Steve Squires, who is the PI on the MER rovers, is also a part of this team. The ability of astrobiologists to work with the likes of Steve Squires has been very instructive to us in how we can design technology on Earth and think that it's perfect and we know everything about that, and then you take this off and you try and get a rover to do what you could do normally is definitely a challenge. And what I have on this final slide that's coming up is just showing where we are in this whole procedure, and that is actually taking a rover and integrating this with instruments, which turns out to be a very hard thing. First, you have to have the intellectual integration of not being able to just pick up the sample, but then you have field testing of instruments, and are they cold ready? Can they work under harsh conditions? We've made it through that step. And now we're integrating things with the rover, and in the next 3 years we're going to take out a more substantial rover. We're working with Pan Conrad at the JPL on native fluorescence instruments. With the SAM team, that's Sample Analysis of Mars, Paul Mahaffey, who is PI, is working with us, and Jen Eigenbrod, also from Goddard. So this is inter-team collaborations. And finally, Dave Blake, who is the PI of Kemin, comes along with us up to Svalbard on the Amaze expeditions. So that wraps up the biology portion.
All right. So I'm going to conclude this. If I get this to move. Let's see. Aha. Okay, there we go. There we go. All right. So this last slide is always a dangerous slide. As Dante Loretta just told me about 3 days ago at the NRC, that graphology is always subject to lots of interpretation. But what I'm trying to show in this slide is that, in fact, we have a very interdisciplinary team, and overall we have a very, very well-balanced team. So if you think of TACS 1 and 2 as being astronomy related with a lot of crossover from people like myself working with people like Alicia, you'll find that it breaks out to about 12, 12, and 12. And this is actually a very, very conservative graph. In fact, I've missed Alicia in past 2, so it's just we get called to go a little faster. But if I were to make cross links to where people's interests are and where our discussions lead us, it would in fact almost be a full field. So that's that. And the last thing, you may not be able to see, because I can't see it, is the majority of the funding is directed towards the support of postdoctoral scientists at CIW and other institutes. So much of what you see is the science that we do, and what our grant is actually the funding is directed towards the support of young people who we've had tremendous success throughout the years. And I don't know if you can see this anymore, so I'll conclude there.
You can see it.
You can see it. Okay, we can't see it. All right. Well,
windows up here.
so that's the essence of it. I guess I would just say that Matt Schrank is a classic example. Julie Huber's another. Matt we funded in Can 1 as a student. John Barros's. He was an NEI NRC postdoc in Can 3, and now he's a co-I in Can 5. And the number of postdoctoral associates that we've been able to support through this has been spectacular, and they're now across the country in many, many major institutions. Dave Johnson was supported through our NEI Can 3, is now a young faculty member at Harvard. So what you're seeing in terms of what the proposal looks like in a budgetary sense is actually a very strong educational component. And we serve as mentorship in a broad array of astrobiological problems. So I'll stop there. Do you have any questions? Be happy to.
Okay, George and all the rest of the speakers, thank you very much. I think this worked out extraordinarily well. You guys did a fabulous job putting this together. We do have some time for questions, but I would encourage you all to communicate with the investigators on the Carnegie team by email, etc. As you know, the purpose of these seminars is to enable collaboration and integration across the Institute. And so feel free to email the Carnegie investigators and their colleagues and everybody who's either been speaking today or whose work has been described. And we encourage all the interactions possible. And right now we'll throw it open for some interactions in Q&A. You can either raise your hand in WebEx and Marco will call on you, or just break in right now. Do we have any hands raised? If anybody has a question, why don't you just open up your mic and ask, and then we'll call on the others by WebEx.
And if you don't have a question, that's okay too.
No, it's not. We like to demonstrate interactivity in these, George.
Okay, Carl. We'll sit here until somebody asks a question.
Somebody ask.
Don't be shy.
Okay, Lauren has a question. Georgia Tech.
I'm trying to figure out how to get my am I on mute? Can you hear me?
I can hear you.
We can hear you.
You can hear me. Okay. I wanted to ask George about his little nodules, those organic nodules he found, and ask if he has figured out the molecular weight of these polymers, and if he's tried to do mass spectrometry of their fragments and that sort of thing. I was very interested in these polymers.
Yeah, we are too. We've only just literally these things are fresh off the press. We just discovered that a lot of what formaldehyde. I mean, others have known, and this is why we were terribly surprised to explore this formaldehyde question. But precisely what controls the nanoscale formation of these little spheres is not known. And I've had an opportunity to talk to.
Just the chemistry. What do you understand about the chemistry?
The chemistry, I think I understand quite well. If I were to give it in a series of steps, what you're looking at is sort of Formose-type condensations, progressive elimination reactions, all in sort of a very random sort of system, tautomerizations, Amadori rearrangements. But the key seems to be Diels-Alder cycloaddition reactions that start to allow to build up a three-dimensional network. So they're completely insoluble. So they're not very amenable to mass spectrometry. So now, ironically, to study them further, we have to find ways of now taking them apart in a systematic way. So that's what we're doing.
There are no more hands raised in WebEx, but if you have a question.
Yep. It's Mike Luma. I just had a question to follow up on George's comment. I've been sitting here thinking about these hollow spheres myself and wondering how the heck do you get a hollow structure in something as processed as meteoritic material? And in particular, if it was preceded by some icy body or was an icy matrix, what kind of a sequence would lead from that icy matrix towards a matrix that had hollow spherical inclusions?
Well, I could give you my opinion, but maybe Larry wants to chime in.
Let's hear George's.
Well, we don't know. Obviously, that Fib image, that beautiful focus ion beam section that Rhonda Straub made, and that we've analyzed 6 different ways to Sunday, as they say, caught us all by surprise. She just did a random section through an ordinary chondrite, and there in the center was this little organic donut, as you observe it. Of course, it's a hollow sphere. So it begs the question whether it was always hollow, whether there was a fluid that once was in the core of this material. But the shape of the thing and the fact that Keiko Nakamura Messenger has been studying this for quite a bit, and in the high resolution, you almost start to see concretionary features. So my tendency is to look at these and think they scream out as products of aqueous processing. But then Larry Nittler, who doesn't want to talk about this right now, could tell you something about the isotopes, which say that may perhaps a very different story.
Well, I guess... What?
Huh? Yeah, Larry's right there.
Oh, I'm somewhere in there, I guess.
Here, I'll get you.
No. What we're finding with these is, one, they're a very diverse set of things, that they're not all hollow. Some of them are solid. Work by Lawrence Garvey at Arizona State doing fibbing of these things is showing that some have multiple little holes within them. They're not just necessarily single hollow things, but many of them are. We're starting to get good chemical data, carbon X-ray edge spectroscopy on many of these globules to compare with the surrounding material in the IOM. And in any given meteorite, they seem to bear a lot of chemical similarities to the general IOM of that meteorite. They're not all one population that's mixed in with the different meteorites. Their processing seems to be related to the overall processing of the meteorites. And they're typically isotopically quite anomalous relative to the other organic matter. Even though they're chemically similar, they are typically more enriched in deuterium and nitrogen-15. And all of this has to go into finding an explanation for them, but we don't have such an explanation yet.
But I assume they constitute a tiny fraction of the total organic material, where IOM is the dominant fraction.
They actually, I don't know about dominant, and that this is an extremely important question, but it's a non-trivial amount, especially in a couple of the meteorites like Tagish Lake and Bells, which is an unusual CM chondrite. Both of them, it's a major fraction of the IOM is made of these little globules.
I would say that Rhonda sectioned randomly through an ordinary chondrite and picked up one just like that. So that's one sample, but still the statistics must tell you there's a fair amount of them there.
So is it correct then to categorize this as saying that the most primitive cometary material has the highest fraction of these nanospherules?
That we don't know. We have found 2 of these nanospherules in the Stardust, in the cometary Wild-2 samples. But how representative... Huh?
Oh, but don't you regard Tagish Lake as cometary material or close to it? Why not?
I don't know of any evidence that would link Tagish Lake to a comet.
But certainly is far more primitive than most other CIs and CMs.
No, it's actually more processed than some of the other carbonaceous chondrites. It's very primitive in some respects, but its organics are actually somewhat processed relative to those in other meteorites.
We should have you come and give a seminar.
I'd be happy to.
Good.
So Chris, did you have a question?
Yeah, we had 2 questions. First, staying on the organic matter question from Larry, we were curious about how often the deuterium and the N15 hotspots are correlated versus non-correlated. It looked like sometimes the N15 and the deuterium is the same spot, sometimes it's not.
Yes. No, it's a good question. I don't have a way to quantitatively answer that in simple sentences. What I would say is we see correlations and we see anti-correlations in this material, that some materials have both, some have one, some have the other, and there's no simple correlation.
Okay, and our second... Oh, sorry, go ahead.
No, go ahead.
Okay. Our second question actually is about Methanostar Sinales analysis. So if Barros isn't around, it may not be answerable. But we were curious, the slide said 90% similar to Methanostar Sinase species, and we were wondering how much that would drop off if you went to the 95% level or something, because that's a pretty low cutoff.
I think it was 80%, wasn't it? In fact, I thought it was 80.
It was 800 were within 90% of 800 clones were within 90%, or 800 sequences were with 90%, or something like that.
Sorry, here we go. 800
species.
Oh, okay. Those are the OTOs. I wouldn't know the answer to that. You'd have to. But you can email John Barros. That'd be a great idea. Or Julie Huber, maybe.
Yeah.
Sure, no problem. Okay, thanks.
Hi, this is Sean McGlynn at Montana State. I've got a question concerning some of Robert Hazen's work and the idea of the mineral evolution. And I'm curious about if we plot different planets and different atmospheric compositions and the mineral diversity on those, is what we see, I think there were 4,300 different mineral species on the Earth. Is that really just a side effect of oxygenation, or is there something else going on?
No, it's very much. I can't. Bob's not here, so I'll try to answer for you. Yes, if you go into his paper, he would argue that coming through the Great Oxygenation Event led to, of course, a very broad distribution of highly oxygenated species that wouldn't exist prior to that. And so it also changes the weathering characteristics of the continents, if I got that right. So one could reasonably assume that the changes of that proportion of atmospheric chemistry would have profound effects on essentially the weathering cycle and the recycling chemistry associated with it. It could, in turn, it'll eventually start to change the chemistry of the mantle, right? So, does that answer your question?
Yeah, thanks. I'll check the paper out.
Yeah, check it out.
Okay, well, I would like to thank everyone who participated in this once again. I think this Q&A just illustrates the NAI process at work. So thank you all again. We will be hearing on Wednesday, same time, same channel, from Dave DeMarais, who's sitting right here with us at NAI Central. And then we'll be continuing on. So thank you all again. This has been very, very effective. And thank you particularly, George, and Marilyn, Alicia, Steve, for all your preparation, and George also for zooming in the camera on the speakers. I think that was just an exemplary way of really making the personal connection. So you did a fabulous job. So thanks, everybody, and we'll see you on Wednesday. So long for now.